Again, thank you very much to the EMA organizers. It's been an exciting day, a very good discussion and debate. I entered this talk on clinical development issues in progressive MS, thinking a little bit about our location here. So on the left side, you see a more historical picture of the region that we're in London right now, and here the more modern day view. Also, I believe this is the Tower Bridge. And it reminds me, as the last speaker of today, to assure you with the words of Henry VIII to his late wives that I shan't keep you long. A little bit of history of the multiple sclerosis phenotypes, maybe as they pertain to the progressive forms of the disease or to progressive MS. This is from McAlpine's 1972 second edition of multiple sclerosis. And as you can see, there are 7 phenotypes, and there are some forms that we no longer recognize in the classification today, but they're clearly phenotypes that all of us as neurologists have seen in MS patients, and there are probably some phenotypes that we have seen, as I was discussing with Professor Ebers, that are not on these 7 phenotypes. So the question really becomes at this stage of our modern understanding of the pathology of multiple sclerosis. Is it one disease? Do we have 7 phenotypes? Do we have 4 phenotypes of the existing Lublin-Reingold classification? Or maybe we should go with the in-press, I guess, new Lublin-Reingold classification that dropped apparently the progressive relapsing form to 3 phenotypes. And I'm running into the same issue. Yeah, I'm just too much animation. Thank you. So the question really is, for my brief expose here, how useful are the existing MS phenotypes in guiding clinical trials for approval of drugs in progressive forms of MS? And so I will take the position of the gentleman on the right, the lumper, in the pathogenetic and etiological heterogeneity that we're all encountering. The splitter would be to the left and the fence straddler in the middle. And I've illustrated this also with this nice package of paraphernalia found between 2 cardboards. This would be the splitters, looking at the package this way, and this would be the lumpers. So I'm going to summarize my talk, and then I'll give you the evidence for my contention and Teva's contention, which is a case for progressive MS. I know this is a term that Alan Thompson has used for a decade now for his group of progressive multiple sclerosis. MS is a chronic inflammatory disease at all stages of the disease. It affects the entire CNS, not simply the focal demyelinating plaques. And we've seen many examples and evidence for that today already. Many different immunological mechanisms lead to demyelination in MS, and subsequent neurodegeneration is associated with activation of microglia and astrocytes. MS is biologically one disease. When adjusted for age and disability status, there are no differences found between the phenotypes in epidemiology, in age of progression onset, in the genomics of the disease, in the genetics in familial forms, and in MRI brain atrophy rate, as well as in T2 lesion load and distribution, if appropriately adjusted for age and disability status. Preventing and delaying disability progression is the recommended primary endpoint in PMS studies. That is our recommendation. EDSS measurements are currently the primary endpoint of choice, although we all recognize, and especially after today's discussions and elaborations, that it is a suboptimal endpoint. We believe that less relevant are the effects on superimposed relapses and selection of patients based on acute active lesions, as the DMT's effects in this axis of the pathology have been well established. That is notwithstanding Gordon Francis's comments that, of course, in the selection of patients, you will encounter a certain percentage of people that do have relapses, but the effect of the appropriate DMT, disease-modifying treatment chosen for progressive MS, should be, as Gordon Francis stated, should be stratified, or there should be a sensitivity analysis so that we can distinguish in these trial populations the effects on suppression of the relapses versus the effects on the underlying progressive illness. And so the key comments from Teva, not part of the EFPIA, on the EMA guidelines are summarized on the slides. The current science does not support clearly discernible genetic, pathological, or epidemiological clinical features which differentiate RMS that leads to SPMS from PPMS. Both of these progressive forms of multiple sclerosis are united by age of the onset of the progressive form of the patient. The recommendation, therefore, is to enable the study of both phenotypes of progressive multiple sclerosis in one study population for an appropriate disease-modifying treatment that has a mechanism of action that can be located into the CNS and is likely related to affecting the neurodegenerative process rather than the acute inflammatory lesion process. So this should be done in one combined patient population to address the uniform disability progression, to enable more rapid recruitment and to enable more efficient clinical trials of this dire needed treatments in the progressive forms. And as was said earlier today, and so we don't have to wait every 15 years to hear about another failed study. So the inclusion should be done by age, and they should be very carefully included by age criteria and by EDSS criteria. The question of oligoclonal bands, as Professor Giovannoni will point out, is still a question of discussion, especially for the PPMS population, as there might be somewhat differences in the progression rates within the PPMS. The primary endpoint of such a study should be the rate of disability progression. We would advocate to use both the EDSS and the individual modified MSFC components with the Ralph Benedict adjustment of substituting the PACET with the SDMT, but retain the time to walk, which was discussed eloquently today as a very useful tool, and also carefully assess the 9-hole peg test, potentially with a higher percentage of change. But this would be a combined endpoint. I think it's very similar, if not identical, to what was presented by Biogen earlier today by Gilmore O'Neill, the EDSS plus. And there's one further comment, not in the topic of my talk today, which is that in RMS, the primary efficacy parameters should be interchangeable. That means for the appropriate treatment, progression should be equally considered and interchangeably considered as a potential primary endpoint to relapse rate and not necessarily requiring a co-primary endpoint. So here, this is the lumping of the clinical course, the natural history, and the epidemiology. And my task has been made very easy because I believe there's not a single slide in here that hasn't been shown already today, so I can refer to this in a new context. This is a new slide. This is from Helen Tremlett's group on the British Columbia database. It really shows very nicely that the time to sustained and confirmed EDSS-6 by disease course from time of onset is quite different between the relapsing phenotype and the primary progressive phenotype, and the difference is astounding. It's a 17-year difference in the median time to EDSS-6, and this is probably one of the reasons why these phenotypes have survived. When you look at survival, which is the ultimate equalizer, from birth, these two forms become identical. This is what's meant by these are unified by age. The progression onset is at the same age between the relapsing remitting to secondary progression as the primary progressive MS. And if you look at this, at the survival from MS onset, then it appears that the RRMS phase is skipped by those that start with PPMS. And these are just the numbers confirming this. Again, supporting the hypothesis, it's the same biology, the same disease with different phenotypes. We've seen these slides from the London, Ontario Scalfari paper several times. I'm just trying to make the point that the total relapses, they may of course be confounded by several things, but the time to progression seems to be rather uniform. And I'm going to skip the subtlety of the year 1 and year 2. and again, talk more about the fairly uniform progression to EDSS-6. This is this paradoxical finding or pseudo-paradoxical finding that those people that continue to have relapses are not progressing. An important paper that maybe hasn't been looked at today yet is here by, also by the Biogen group, Gilmore O'Neill and Diego Cadavid in the audience. It shows that when you look in a clinical trial setting, which is really the topic for today, that SPMS and PPMS study cohorts, patient cohorts, are behaving identical with the exception of some minor variations in the 9-hole peg test and one EDSS sensory measure. Otherwise, in these two trials, there was identical behavior on all clinical endpoints. This slide was shown before, again, to show that PPMS and SPMS have the same time course to hard endpoints. And I know that the key people from the Cupid trials are in the audience as well. This is a point of subtlety that we're facing when designing studies in progressive multiple sclerosis. Here it occurred that there was large over-recruitment apparently in EDSS-6 and in EDSS 6.5, which translates to low progression rates. And it almost appears that by, this from the Cupid trial, that it almost appears that by strata of these EDSS groups at baseline, you can predict the progression rates, with the highest progression rates seen in the lower end of the included patient population. So a careful balance between age and EDSS inclusions is needed in order to enable this proposed PMS trial population to study the effects of disability progression. Just a little bit of evidence from the genetics and on the phenotypes. The cumulative genetic risk from genetic studies and from familial studies have not been able to identify differences between the phenotypes. These investigations, given the available technology of genetics these days, are ongoing. We very recently collaborated with the San Francisco group on a very large PPMS genetic database that Teva has. They thought they had a lead on a gene signature for PPMS, which then was falsified by the PPMS data set. So these are ongoing investigations, and time after time, investigators have failed to find any differences between the phenotypes. This is a very telling publication, maybe usually read in a different context. This is the family tree of a pseudodominant MS pedigree containing a TYK2 mutation. TYK2 is part of the obligatory interferon signaling pathway. And in this pedigree, all MS phenotypes were encountered with one single mutation associated with MS in the pedigree. When we look at the cumulative genetic risk for multiple sclerosis, it's clearly identical between RR and PPMS, and of course different from the controls. So from a genetic perspective, these two phenotypes are identical. And when we look, this is Jack and Telt's review in Acta Neuropathologica. When we look here at the GAD enhancement, which is not matched for age, of course, there seems to be some more gadolinium enhancement in the SPMS. Again, this may be the transitional period that Gordon Francis was referring to, rather than the PPMS, which seems to be more strict in its diagnosis and not allowing for GAD enhancement too much, at least, on the phenotypical description. When we look at black holes, there seems to be a slightly higher tendency in the literature to report black holes and more atrophy, slight more atrophy. In PPMS, again, I think these are variations in forcing patients into the phenotype. When we look here at the atrophy in the spinal cord, maybe the common pathway, at least for the ambulatory disability, that is both strongly affected. This slide has been shown now for the 4th time. The point I want to make is that the atrophy rates occur early, and the rate of atrophy is actually consistent between all phenotypes, including CIS. Again, speaking to the biological organic nature of the disease and speaking to the fact that the accumulation of T2 lesions and the accumulation of inflammation does not seem to be altering this underlying atrophic process. So there's extensive cortical demyelination in progressive multiple sclerosis. And when you really look at the cortical lesion area between PPMS and SPMS, those are identical with regards to the effect. There's a difference in the white matter lesion area, and again, this may be confounded by diagnosis. Microglial activation appears, at least according to some neuropathologists, the common feature in progressive MS. I've just highlighted in this unwieldy graph for you the active white matter lesions, the slowly expanding white matter lesions, and the inactive white matter lesions, and again, very similar quantitative results in SPMS and PPMS. And the microglial activation is the unifying underlying pathology, according to some. This is from the Lassmann group in Vienna, with high scores of microglial activation in both progressive forms of multiple sclerosis. And again, from Jack and Tell, the histopathological findings show, by and large, identical patterns in summary of the literature between PMS, PPMS, and SPMS. And if there are any differences, they are slight, quantitative, and not qualitative, and can be explained by biases in the diagnosis towards these slight differences. So what is going on? We have, of course, an inflammatory disease, autoimmune disease that gets established in the CNS. There's inflammation with new waves of lymphocytes entering the CNS. There's a profound, at times, blood-brain barrier disturbance. And we have classical active lesions that are highly associated, as we've learned from Maria Pierre-Sormani's work, with relapses. And then we have, at the progressive phase, we have a trapped inflammation, to use the term that Hans Lassmann likes to use. We have some people find meningeal inflammatory aggregates, others cannot find those. We have slowly expansion of pre-existing lesions. We have subpial cortical demyelination and diffuse white matter injury. And inflammation is the trigger, potentially. The activation of CNS resident cells, microglia and astroglia, are very likely the secondary effect that then leads to oxidative bursts, liberation of iron from cellular stores, mitochondrial injury, and then to the oligodendrocyte, axonal, and neuronal injury. So duration of disease and age are accompanied by a change in the pattern of inflammation in the brain. The inflammation with activated microglia and astrocytes persists in the CNS behind a repaired, largely intact blood-brain barrier, and we would require potentially a different DMT MOA compared to RMS. So in summary, there's no genetic differences, there are no familial differences, there are no pathological differences, and any of the differences seen on MRI or immunology can be explained by other biases. So the current tools for measuring progression in MS are suboptimal. We've agreed all on this, yet we probably are going to be using John Kurtzky's scale for quite a while in drug development for progressive MS. And we would advocate to replace the base set with the SDMT and allow for the individual MSFC components to contribute to the endpoint. We are very keen on using biomarkers to further establish the efficacy and the effect within the CNS compartment. And I think those are effects that will help us with robustness. We believe brain atrophy will be an important biomarker of these effects. And so the suggested key enrollment criteria for such a progressive MS study would be McDonald criteria, SPMS or PPMS, evidence of clinical disability progression in the prior 2 years, no relapses in the last 2 years. The age range would be between 25 and 55 to avoid over-recruitment of patients that are probably not going to be responsive to a progression-modifying therapy. The EDSS range can be fairly wide, but it should definitely be curtailed at the higher end to not run into low progression strata problems. GAD at baseline should probably not be allowed, but I put a question mark. And of course, there should be normal organ function. I've read this slide to you in the beginning. These are Teva's recommendations. And I leave you with another image of the area. And thank you for your attention.